Hello and welcome to Allied Health Care. So today we are going to discuss about a special media, that is HiChrome Agar. Nowadays we are using HiChrome Agar for the determination of bacteria that can causes UTI. Previously we have used this type of media, that is MacConkey Agar. MacConkey Agar produces two types of colony. One is lactose fermenting colony and another is non-lactose fermenting colony. But there is a limitation. We can identify whether the bacteria is lactose fermenting. Lactose fermenting means this colony produces pink color. Here we see that the colony is in pink color. If the bacteria is non-lactose fermenting, then the colony appears as pale. So we can only identify whether the bacteria is lactose fermenting or non-lactose fermenting. And another thing is, this is MacConkey agar, that is selective agar, and it has bile salt, so it can restrict the growth of gram-positive bacteria. So we just understand whether the bacteria is lactose fermenting or non-lactose fermenting. Furthermore, we have to do different type of biochemical test to identify the actual species or bacteria. So this is very laborious, time taking or time consuming to identify a bacteria. If we inoculate in the MacConkey agar, then we can get only lactose fermenting or non-lactose fermenting colonies. Furthermore, we have to do biochemical test. So HiChrome agar can replace the MacConkey agar because there are different type of colony with different color can be produced in the HiChrome agar. The HiChrome agar, the composition of HiChrome agar contains different type of chromogen material. It has chromogenic mixture. So different bacteria can produces different type of colors. So if we understand this type of colors and if we learn which bacteria causes which color, so there is no need of biochemical test. We can identify the bacteria merely seeing the colony. So we can replace the biochemical test. We can replace the biochemical test and we can identify just seeing the colony of color in HiChrome agar. But it is not mandatory that we do not do the biochemical test. Sometimes if confusion arises, we have to do the biochemical test. So now I am demonstrating the different type of colonies. First of all, I am showing the E.coli. So this is the colony of E.coli. You can see the color of E.coli is purple or magenta. The purple or magenta color demonstrate the bacteria, that is E.coli. Actually, the E.coli produces beta D galactosidase. This beta D galactosidase can cleave the chromogenic agent and it can produces this type of magenta or purple color colony. So it is E.coli. Further, if we corroborate or if we confirm the bacteria, we can do indole test. If indole is positive, then the bacteria is 100% E.coli. So this is the another colony, that is Klebsiella. Klebsiella in HiChrome agar, Klebsiella produces slightly mucoid colony, or you can see the color of the colony is metallic blue. If we get slightly mucoid colony with the color of metallic blue, then we can identify this bacteria as a Klebsiella pneumonia. So here you can see the two species, the two sample is identified as Klebsiella. So the color is metallic blue. Next, from this colony we can see there is a numerous small colonies. And this colony is identified as Enterococcus because the color of the colony is blue. You can also see the picture of actual picture of Enterococcus, that is blue color. And there are several other type of bacteria, as you can see from the picture of the left corner. That is, there are different type of bacteria. First one is Proteus, which produces brown color. Enterococcus, that I have already told that it produces blue color. Another one is Staphylococcus aureus, that is golden yellow color. Candida, light green color. And Enterobacter, that is pink, that is produced pink colony. So by using HiChrome Agar, we can replace the necessity of MacConkey Agar. And another one colony I am showing you, that is Pseudomonas. The Pseudomonas colony produces no color. There is no color produces. So we can see the Pseudomonas is pale color, or there is no color. No color is produced at all. How can we identify the Pseudomonas? Do gram stain. It will be gram negative, obviously. And another one is the confirmatory test for Pseudomonas is do oxidase test. As we see in the picture, the oxidase test will be positive. If oxidase test will be positive, then the bacteria is identified as Pseudomonas aeruginosa. And another interesting thing about this type of colony, this type of media, here in this media different bile salt is incorporated, so that this type of media, just like MacConkey Agar, can inhibit the growth of gram positive bacteria. It can only allow the growth of gram negative bacteria, just like MacConkey Agar. MacConkey Agar, we know that MacConkey Agar contains bile salt that restrict the growth of gram positive bacteria. It can allow only the growth of gram negative bacteria. Here, in case of HiChrome Agar, only gram negative bacteria growth will be allowed. Gram positive bacterial growth will be restricted. So in this lecture, we can understand the HiChrome Agar, and we can demonstrate how to identify the different type of colonies in HiChrome Agar. So stay tuned to our channel Highlighted Educare. Subscribe, share, like with your friends. Thank you very much.